Good afternoon, everyone. So Sergio gave me an introduction. I actually used to work in Europe before, so it's always nice to come back. Last time I was in Prague was a couple of years ago in November. It was cold and dark and rainy, so it's good to see this weather. Very similar to San Jose weather. So I've just put a little sign of San Jose there. I moved there about 10 years ago. It is the capital of Silicon Valley. This is where we design and make the triple quadruple instruments. This is where Bob Finnegan started his original company, and then eventually he merged with the company in Bremen, and we became Finnegan MAT. And now today we are Thermo Fisher Scientific. But this is where it originally all started, and we still work out of the same building, which is nice. Bob still pops in occasionally. It's good to see him. So what I'll do in the next 30 minutes or so is give you an overview of all our LC-MS products, which are used for pesticides analysis, can be used for veterinary drugs or other contaminants as well. This is just going to be an overview. There's no way I can go into any depth, but my colleagues as well as some of the other eminent speakers will go into it in a lot more details later on today and tomorrow as well. So I showed this slide last year. I mean, this is something that you are all familiar with. We're looking at mycotoxins, PPCPs in environmental, and of course, we're focusing on pesticides. I took a picture of this when I was in DC, in Washington, DC. This was an advert on a bus. 70% of US antibiotics go to animals that aren't sick. So you wonder, how are these controlled, et cetera, even in the US? And my role is a global role, and trust me, when I go to some countries outside Europe and North America, I see a lot of stuff being used extremely legally. And so this is one of the reasons why we have to monitor for this, as well as the targeted stuff. We have to look for unknowns as well. This reminds me of Sudan dyes. Sudan dyes are dyes used in shoe polish. I'm wearing red shoes today. This is typically used, but it was found in paprika in Europe. So it's quite risky. Whatever we eat these days, it seems that there's some illegal substances in this. And of course, the PFOS, PFOA that you are aware of. So these are SOP-driven techniques used by triple quads, primarily LC-MS or GC-MS. But what I'm seeing in the industry is what else could be present. When I look at the overall environmental and food safety market just for LC-MS, even today I see about 70% of the instrument sales are based on triple quadruple. So everyone is still doing a lot of targeted work, but the amount of unknown screening is definitely increasing, and it has changed a lot in the last 5 to 6 years. Now, one thing I want to introduce to you here is that we started off using HRAM, say 3-4 years ago, even 5-6 years ago when the Exactive was launched and with the Q-tops for doing unknown screening. But we're seeing more and more people using, especially the Orbitrap technology for doing quantitation as well, and I'll show you some data based on this. So in any analytical methodology, you have the sample prep, you have the chromatographic separation, followed by mass spec detection and data processing. So this is the part that I want to concentrate on. So I'll start off with this slide, and I really would like to get some feedback from you guys. This is evolving. When I finished my PhD, the first mass spec conference I went to was in Paris. It was a OECD conference, and everything was done by GC-MS. And people are saying LC-MS, shows how old I am as well. So it wasn't an accepted technique. And then I started my professional career with Dow AgroSciences, and I started using triple quadruple to do residue analysis. So this was a fascinating paper that Kurt Brunner wrote back in 1987. What is the ideal mass analyzer? Does this exist? Because we have all, we have all these different options, right? And so the market is evolving, as well as the regulations. So we think we have the industry's leading portfolio of MS solutions. I'm again talking about LC-MS here. So on the top left here, this part of our business has grown explosively over the last 5 to 6 years because customers have loved this technology. And now we have the GC Orbitrap as well coming out, this ASMS. So even though I call it the Exactive, this applies to our Exactive platform, which includes all the Q Exactives. Let's not forget this. Triple quads, as I said, used for routine quantitation, still about 65 to 70% of the total market. Iron traps, I've seen this market evolve and then plateau out. Once upon a time, Thermo or Finnegan dominated about 95% of the iron trap business. Today, it is in sharp decline. Okay, so because newer technologies have come out, this technology has taken over a lot of things. And then, of course, we launched the Tri-Brid Orbitrap last year. That basically contains every single type of mass analyzer all built into one. Everyone wants one, but it costs $1 million. So that is a phenomenal instrument. You can go up to resolving powers of 400,000 plus. This is the other question. When or where does resolving power eventually end? Whenever I talk to my colleagues, Alexander, especially in Bremen, he's working on newer stuff as I speak with you today. Right? Next time I speak with you, we'll probably have a new instrument out there. So focusing on the triple quadruples, we have two systems. I just wanted to highlight this. These are used primarily for routine quant work, TSQ Endura MS and Quantiva. Quantiva gives the highest performance in terms of quantitation, and the robustness is very similar on both. Now, one thing I wanted to remind people, I'll have to go on to that slide. This slide is an overview of our Exactive and the Q Exactive portfolio. So tomorrow, Maciej Romirsky, my colleague from Bremen, will go into in a lot more depth about these products and their capabilities, but especially the Q Exactive focus. I don't know how many of you know about this. This was launched last October. It's still relatively new. It's an incredibly powerful instrument. We call it the baby Q Exactive, for the time being at least. It's very similar, and what we are seeing is that it has incredible quantitative performance as well. And again, I'll show you some data later on. Trust me, for the kind of work that you guys are doing, I'm assuming it's mostly small molecules work, pesticides, etc. This is probably too high-end for this. This is primarily used by our colleagues in proteomics. So when I look at the overall portfolio that we have the raw sample, then we have sample prep. Since the acquisition of Dionex, we have very powerful sample prep capabilities with the Dionex A system. We have UHPLC systems as well. We have this product called Econ Max Plus, which offers online sample prep. If you're doing pesticides kind of analysis using water samples, then you can do screening with Orbitrap as well as Orbitrap Fusion, and then quantitation using the triple quadruples. And everything is pretty much driven by TraceFinder. So later on today, my other colleague, Ran Choudson, will take you through the capabilities of TraceFinder in a lot more details. And this is how we actually pull all the instruments in together. So this is one slide. I just wanted to give you an overview of the different scan functions and different softwares that we use. So I was actually talking with my group last week, and we decided to summarize everything on one slide. So if you're doing quantitation or targeted screening or non-targeted unknown screening, obviously the triples play a huge role here. But as I've mentioned already, especially using the Q Exactive, when you can do pre-selection using the quadruple that you use in the Q Exactive is exactly the same quadruple from one of our triple quadruples. You can do extremely powerful quantitation as well as screening. Now, for targeted screening, we of course have the Exactive, but the Q Exactive portfolio as well. And then for non-targeted screening, the Q Exactive series. For scan modes, you would be using the SRMs or MRMs, as also called. The quantitation you can do by full scan, but also you can use, I've seen this. Especially when we started working with customers who are used to operating triple quadruples. The Q Exactive is an extremely powerful instrument, and then it takes a little while to get used to the different scan modes, because there are a good few different scan modes. And again, I'm sure Maciej will talk about this in a lot more details tomorrow. So you have full scan mode, you can do product reaction monitoring, you can do data-dependent MSMS, you can do VDIA, and Hans Moll, who's sitting there, I've worked with him a lot as well. He'll be talking about this tomorrow. He's explored this in a lot more details. And then you can do library matching as well. For targeted screening, you do full scan data-dependent MSMS and these scan modes, and then similarly for non-targeted screening as well as the unknowns. And as far as software is concerned, TraceFinder is the engine which drives all the Thermo it used to be the LCMS platform, but now the GCMS as well. So every single product, mass spec product that you buy from us, they're driven by TraceFinder for both data acquisition as well as data processing. In addition to that, we have Compound Discoverer. Again, Franz will talk about this in a lot more detail to help you with your screening needs. Sieve is a software which was originally developed for proteomics, but it allows you to do principal component analysis, and it's a plug-in with TraceFinder. So if you have a complex matrix, you want to do principal component analysis, you can export the data from TraceFinder into Sieve, process the data, then push it back. Though the latest version of TraceFinder, which is coming out at ASMS, which is the TraceFinder 3.3, will take into account all the major features of Sieve, and eventually Sieve will be pulled out of this. And then we have the spectral library that I'll talk about in more details later. So typical LC triple quad workflow. Again, I'm not going to go through this in details. You know about this much more than I do. So you have the extraction, LC-MS, MS. I will mention this a little bit in the next slide, identification followed by reporting. For our triples, what we've started doing is that we have started offering built-in methods, whether you're doing pesticides analysis, vet drugs analysis, mycotoxins analysis, et cetera. So you can have this in 3 separate ways. We have this software called TraceFinder, into which we have listed all the major SRMs, so you can select those to build your method, or selected pre-configured method which are built into the instrument page. So for example, at ASMS, we'll be launching a pesticides bundle. We're calling it a Pesticide Explorer. So 4 bundles will be offered, one with the Endura, one with the Quantiva, one with the QE Focus, one with the QE Focus for Quan and Qual, for which a 400+ pesticides method is included as a built-in method. So whether you're doing screening or quantification, you can use that. And then finally, you can actually use that method and edit it if you're not going to analyze all those compounds. So we're trying to make productivity easier for you and to be more efficient, especially in the routine quant labs. Now, the triple quad product line that I mentioned were launched almost 10 years ago. And I wonder how many people actually remember what HSRM is. This is something we promoted aggressively when we launched this, because the quadruples that we use in our triple quadruple are very different from the quadruples that are used by basically our competitors. These are hyperbolic quadruples, and we actually started doing quantification using higher resolving power about 10 years ago, before the Orbitrap platform came along. And I've got a small example, and I just wanted to remind people this capability still exists. So tall tetrazool is a substance which is used to treat chickens, turkeys, and pigs, and it metabolizes to a number of products, and it is included in the regulatory guidelines, and you can see the parent compounds as well as the metabolites here. Now, if you look on the left-hand side, when these are quantified, some of them are found running the SRMs at 0.7 full width half maximum, which is the unit resolving power, but these 2 compounds are not found. This is in chicken matrix or other meat matrices. When we took the same sample, without doing any further sample cleanup, and ran this at HSRM, which is the high resolving power, ran this at 0.2 full width half maximum, this was what was seen. Right? So this was when we started doing quantification using high resolving power. So this is offered with both the Endura and the Quantiva, but you get high resolving power capabilities with the Quantiva. So if you're doing targeted quantitation, and if you are running this in complex matrices, this is something which could be beneficial for you. Okay, moving on to the LC HRMS workflow. Again, I'm not going to go through this any details here. You have the identification and quantification or screening here. I'm going to show you some data from our collaborators. I know Professor Amadeo Albert is supposed to be here. I don't know if he's in the audience or not, but his group published this paper last year, and I've extracted a few snippets from that. So this is looking at a big bunch of pesticides after a catcher's extraction, followed by MS analysis, and data processing. I just want to focus on the need for resolving power and mass accuracy. Okay, this is data pulled directly from Amadeo's poster. So if you look at the concentrations here, at 500 ppb, running at 3 different resolving power, you can detect all the compounds of interest. But as you start dropping the concentration, you see a difference. So the QE focus instrument that I talked about, we can run this at 70,000 resolving power, and you can see this. Now, when the matrix gets even more complicated, for example, I forget what this particular matrix was. What's happening is, we've written here is that if you are not running this at the highest resolving power, then you are going to miss out on some of the compounds of interest. Now, Yana showed this, and Katie talked about this a little bit as well. So what we are recommending is that when you're running screening and quantification, you should be running at least 35,000, if not 70,000 minimum. Okay. Mass accuracy, how important is that? So if you look at here, at the legend, for example, we have carbon, hydrogen, oxygen going up to compounds with carbon, hydrogen, oxygen, nitrogen, sulfur, and fluorine. When the mass accuracy is 10 ppm or 5 ppm, you can see a lot of these compounds. But with the more complex compounds, you really need high mass accuracy to identify these compounds. So this shows, again, why mass resolving power and mass accuracy are phenomenally important when you're running complex matrices. In clean water, it's not going to make that much difference. In complex matrices, it's going to make big differences. Again, coming back to my original comment, there are a number of different scan modes. Here's one example from one of our collaborators, again, doing full scan followed by PRM, data-dependent MSMS, and you can see why resolving power of 70,000 is important. This is the product ion, and this is the matrix. They both have very close nominal masses, so you need that. So one graph here from this publication. On the left here, you see the number of compounds detected in full scan mode, but these two actually compare directly full scan data-dependent MSMS and triple quadruples as well. So again, it's showing that it's very similar. One other paper here I just wanted to focus on. So far, I've talked about direct targeted screening and quantification. This was with a collaboration with Christoph Portner and Jochen Turek. They were looking at suspected and non-target screening in wastewater sample. So again, the workflow was very similar. And what we are suggesting, as far if you're doing screening for unknowns especially, is that this is the way to do it. You do the suspected screening and analyze using TraceFinder. You do the differential analysis using SIV, as I'd mentioned. And then if you're looking for specific targeted degradation products, you can use Compound Discoverer. Today, Compound Discoverer allows you to look for targeted compounds, but the next version of Compound Discoverer is coming out, which will let you look at potential unknowns as well. And again, Franz will go through this in a lot more details later this afternoon. At 4:35, I think. Okay, we're going to switch gear now. I know this is written in Chinese. The reason I wanted to show this is that, the biggest need for quantification, and then as Yana mentioned, where somebody asked where our products came from, it's Mexico and Southern American countries. But we also, both in the US and in Europe, import a huge amount of products from the Asian countries. So it's becoming increasingly important that we screen and quantify these. And obviously the governments there are tightening up their regulations as well. So this was a method that we worked on. It's according to the Chinese GB guidelines. Looking at 400 plus pesticides, screen and quantify them as well. This shows a screenshot of the list of pesticides that we looked at. And we did this both using the Endura as well as the QE Focus. I'll share a little bit of the results from the QE Focus. So this was run using full scan data-dependent MSMS with an inclusion list of the 500 compounds that I showed you. All work was done using TraceFinder 3.2, and the confirmation was done using a built-in library that we are now providing. This just shows a screenshot from TraceFinder. This is the quantification, library matching, followed by the calibration curve. And if you look at this calibration curve, it's very similar to what you see on triple quadruples as well. And similarly for another pesticide of interest. This was in beet extract. I want to talk a little bit about the library. We started creating this library last year, and it's now released. Today, we have a combined library for environmental, food safety, clinical, and toxicology. There are about 2,500 compounds in this. This is for LCMS. And all the data were acquired on AQ Exactive Classic, not the Focus, and all data acquisition occurred at 140,000 resolving power. So this is a very clean library, and I've had this discussion, especially with Hans, in terms of what kind of library should the industry be using. You see all the different manufacturers offer different types of libraries. We felt that it is easier if we offered a MSMS spectral library. Okay, so today we have close to 1,700 compounds and close to 9,000 spectra. This is increasing all the time, because we have 5 to 6 spectra per compound running at different collision energies. So whatever collision energy you're running at, plus the step collision energies, you should be able to get a good hit against the libraries that we provide. This is an example. So this was quantified, and this is very similar to the data that Jana showed just before, and confirmation with spectral matching as well. Just want to say one or two words about MZ Cloud. You'll hear us talk about this more and more. This is a cloud-based library that we're working on. This is not fully released yet, but we work with a third party, with Robert Mistrick's group for this. Our aim is to transfer all the libraries from environmental, food safety, metabolomics, clinical toxicology into this. This will become, this already is available. If you go to mzcloud.org, you'll see what's available. But as well as the spectral library, this will also have a lot of metadata included in this, okay? So this is a very powerful cloud-based library that's currently available and is going to continue to grow. So I really strongly recommend, if you're doing screening work, please look into this. We're very, very excited about this. And I think Franz has more details about this. Okay. I'm going to completely switch gears now and just finish off with a couple of slides. And I really would like to get a little bit of feedback from all of you later on today, if possible, because a lot of eminent speakers are in the field who are here today, and I've worked with a lot of you. I am still working with a lot of you and have worked with some of you in the past as well. So if I look at the regulations globally, I've intentionally chosen Japan and China because that's probably where I travel most to in terms of visiting customers. Japan has extremely tight regulations and a well-developed infrastructure. They import a lot of food from China and other countries. The EU is extremely well-developed, as you know. The FDA, I moved to the US about 10 years ago. I was surprised. It caught me by surprise. The EU is definitely a lot more strictly regulated, but the FDA is getting tighter and tighter. The EPA has prescribed methods. The FDA is, they have a lot of methods. They're not prescribed methods, but I don't think they're as tight as the EU regs as well, and then China's coming up. Okay? We have a lot of different types of methods available across these different countries. And this just shows an overview of identification criteria by LC-MSMS according to the different guidelines. And I've intentionally showed this. I think most of the people that I've worked with are part of the authors list here. So I'll not read out all the names there, but all the major people are here. And I really liked that paper. One of the things which was discussed in this paper was the performance criteria. As if you remember the first slide I showed you, the different technologies that we were looking at, what are the identification criteria that we should be looking at as mass spectroscopists? Right? So this was really well detailed here. So this just shows the water testing regulations, again, different regulations, but going towards similar things. So one question I have for you guys, and I don't expect the answers now, is, especially in my role, because I am seeing the tight regulations in Europe, tighter regulations in some other parts of the world like Japan, but other parts of the world, whenever I visit, they ask me, what's happening in Europe? What's happening in the United States? Because their economies depend on exporting a lot of food to the West. So it seems from that paper, I'll just mention you, Hans, but I know most of the other authors are present here. The standardization of performance criteria has been described, and it will continue to be described. We have that. But is it at all possible eventually to standardize any global methods? And I don't have the answer to that. Okay, I think it's something that is worth for us to think about. One of the reasons why I think we get disparity, I used to work, as I told you, before I joined this industry, I used to work in the pesticides industry as a chemist. Back in the UK, when I tried to develop a method and transfer it to the US or then repeat the method back in the UK, sometimes it was difficult. You know that, you're all laboratory chemists. Sometimes it's difficult to reproduce methods between different labs. So is there any way we can actually standardize global methods? I don't know. It's something to think about. Standardize global technologies. This becomes even harder. So when somebody asks me, when I'm traveling in Korea or Singapore, in Malaysia, what technology should I be using? I could say to them, well, you can use this. But that doesn't help that customer who's just coming into LC and mass spec for the first time. So is it something that we can actually help in terms of keeping our food and environmental industry safer, in terms of releasing some guidelines? There's something for you to think about. Your thoughts, perhaps you can share those with me on the boat for those of you who are joining the dinner tonight. Okay, so I'll leave it at that. I'll take any questions if you have any now. If not, feel free to email me. And thank you very much for your attention.